My name is Ryan Hino. I'm a biochemistry major from Santa Monica, California. My project is Nafcillin-Mediated sensitization of Methicillin-Resistant Staphylococcus aureus. So my project is researching how we can increase the susceptibility of MRSA to antibiotics. MRSA is one of the leading causes for hospital-acquired infections, so that has led to a lot more hospitalizations, and it has a very high mortality rate. So we actually have a big antibiotic resistance problem, meaning bacteria like MRSA are not very susceptible to getting killed by these medicines. It's pretty common to just get prescribed antibiotics. You can technically buy antibiotics over the counter in the form of ointments. Over time, the more you use it, overall, the more resistant any bacteria can get, right? It's really just important to have other alternatives, because this is not just a case study or a centralized problem. It's a rising global problem that we may face in the future. My project really started off at the beginning stages, so I had a lot of computational work on the computer, and I'm looking at different genes, which is essentially the genetic code that determines different physical traits. So for us, it would be like hair color, eye color, or height that really drives this sensitization to this antibiotic. So when I came to Oxy, I was really interested in research. So I was reading about all the different research that goes on in the biology department, and I read some of Dr. Okumura's work, and I was really interested with her. So I met with her and started attending some of her lab meetings to see more what she worked with. So working in Dr. Okumura's lab, I typically wake up pretty early to get into the lab and get started. So I walk in and I don my lab coat and my gloves because I'm working with very infectious bacteria. And they're typically 8-hour days. I'll be walking back and forth to different machines, and then by the end of the day, I'm usually planning my next day's work. I grow the bacteria from taking a frozen stock of the MRSA, and then I put them in this special bacterial media to grow overnight inside of this heated incubator that shakes to help the bacteria grow better. I was very surprised to learn that the antibiotic I'm researching with MRSA, Nafcillin, actually leads to this increased killing for the bacteria. It's not really explored much in the literature so far, so I'm really excited to dive more into it. One thing I found different about my project was all the computational or computer work I had to perform. It's very different than the traditional bench lab work that you might see in the movies or in TV shows. So computational research is working with different data sets from biological experiments, so that you can plan a project without actually wasting any time or resources in the lab. There's a lot more reading and writing than I initially thought when I got into research than I do for my project. I consult with my professor a lot to see how to move forward with my project. I think my findings will have a lot of applications, particularly in the hospital. Patients with MRSA infections often are treated with a wide array of treatments and a cocktail of antibiotics. It would be really great to find a more simple and effective treatment to treat these infections. As a science scholar, I really hope that I can just play a small part in helping out the research and medical community and bring a lot more focus to my topic and maybe inspire others to work on it as well.